let's get started then. So today is going to be the 7th week. We are very close to the end of this course, but I hope you enjoy that very much. 1 second, I just have to get this done and we'll get back to you soon. Yep, here you go. So we are in the week 7 again, chemistry in agriculture, pH scale and nitrogen cycle in nature. And the very end, or in the half of this lesson, Dr. Jim will take over to finish this part. So our lesson is pH scale and nitrogen cycle in nature. So let's get started. Our topics today, the pH scale. We're going to cover hydrogen ion concentration. It's very basic. We start with very basic. Acidic, neutral, and alkaline. C, saline and neutralization reaction. D, the pH scale. And we're going to see about the biological fixation, host-dependent and host-independent fixation, nitrogen fixation, and so forth. So let's see some of the slides. And finally, Dr. Jim will talk about the farm management for enhanced nitrogen fixation. So let's start with hydrogen ion concentration. Very basic, very simple. So concentration of hydrogen ion is a measure of the acidity or alkalinity of a substance. The hydrogen ion concentration in solution, a measure in mole per liter, can be calculated using the equation of formula hydrogen ion concentration equal to 10 power to minus pH. For example, the pH of a tomato juice is 4. Then the hydrogen ion concentration equal to 10 power minus the pH, which is 4 in this regard. So it's 10 minus power to the minus 4. So we got 0.0001 mole per liter. This is very basic. So let's go a little bit more practical, but still very basic. Acid, neutral, alkaline. So the chemistry of life is sensitive to acidic and basic condition. So what is acid? So I think we know it nicely. A compound that releases hydrogen ion to a solution is an acid. And a compound that accepts hydrogen ion is a base. So acid is a proton donor. So basically remember that hydrogen ion is a proton, no electron there, only positively charged. So when we say that hydrochloric acid, when it donates the hydrogen to water, it creates H3O plus. So the water accepts the electron and the hydrochloric acid actually donate the electron because it donates the electron, it donates the proton. So because it donates the proton, so we call it is acidic, or the hydrochloric donates a proton, and so it is an acidic. In this regard, the H2O, the water in this case, accept a proton and it's a basic. So acid-basic reaction. So what is a neutral solution? We're going to see, and we're going to cover a little bit about pH scale. In a neutral solution, the hydrogen ion and the hydroxyl ion concentration are equal, and each is equal to 10 power minus 7. A pH of 7 is neutral. So the pH scale describes how acidic or basic a solution is. The pH scale ranges from 0 to 14, with 0 the most acidic and 14 is the most basic or the most alkaline. Each pH unit represents the tenfold change in the concentration of hydrogen ion. Here are some of the example common acids and base. We can see here from the weak acid to the strong acid. The weak acid, acetic acid, we know it very well, hydrofluoric acid, phosphoric acid, and the strong acid are here in this example, nitric acid, hydrochloric acid, hydrobromic acid, sulfuric acid, and perchloric acid, which is the strongest acid in this regard. And we move a bit from the base here, weak base to the strong base. Ammonia is weak base. Calcium hydroxide, considered to be not that strong. Barium hydroxide, potassium hydroxide, and sodium hydroxides. These two are strong bases. So if we compare the concentration of hydrogen relative to the hydroxyl, so we will find three different formation here in acidic, neutral, and base solution. So in acidic solution, and here in the neutral solution, the concentration of hydrogen ions basically equal to the hydroxyl ion, so it's neutral. So when the hydroxyl is predominant compared to the proton, so then we say this solution is base. So just to summarize the difference between acid and base, we see here acid pH less than 7, and as opposed to base pH bigger than 7. Again, acid sour in taste, and base is bitter in taste. Acid is react with metals, base doesn't react with metals. And there are some common things between this acid and alkaline. So they are alike, say like acid and base affect the pH and litmus paper. So acid and base related to proton concentration, and the sum of the pH and pOH equal to 14. So if you say like the pH is 3, then the pOH is 11. So it's the same thing. The measure is the same. All right. So let's move a little bit about the neutralization reaction that create salt. So when we add acid to base as a reactant, acid react to base. So what we get salt and water as a product. So we get salt, which is saline, and this is what we call it the neutralization reaction. For example, in everyday life, when you got stung with bee, the bee venom is basically formic acid. That's why we feel itchy and we feel pain. Because it's acid, we can apply baking soda to that location, and then it get neutral, and the formic acid have no give some irritation feeling to our body. However, the wasp venom is a type of bee also. It's basically alkaline, and we apply vinegar, which is mild acid, then it becomes neutral. So this is in everyday life we can see on that way. So in neutralization reaction, both acidic and basic solution neutralize the effect of each other, and the nature of both acids and bases get destroyed or get neutralized. So here are some examples of the reaction. So hydrochloric acid, when we add with the base sodium hydroxide, what we get? We get salt, which is sodium chloride and water. And another example, hydrobromic acid with potassium hydroxide, we got a salt, potassium bromide, and water. So here are more examples, acid and base reaction. We can pick one more example probably. A sulfuric acid in this case, when it react with calcium hydroxide, it gives rise to calcium sulfate and water. Again, it is neutralization reaction. So let's move now to, yes, we're still in the neutralization reaction, but let's take a look at a video just to see a illustration. To the next part, we're going to cover about the pH scale. So the pH scale indicates concentration of hydrogen ions. And neutral is at 7. So the pH at 7 is the solution is neutral. So the pH scale determined by taking negative value of exponents of its hydrogen ion concentration. So here's the formula for pH. So pH equal to minus logarithmic of the hydrogen ion concentration. For example, if the concentration of hydrogen ion in a solution equal 0.001 or 10 power to the minus 3 mole per liter, then the pH is minus log the concentration of the hydrogen ion, which is 10 power to the minus 3. So we got the pH is 3. So very simple, it's very basic. So here are some of the examples of this ingredient or materials that perform in a different pH scale. So pH, we start from the middle here, where it's not blue in color or it not turn the litmus paper to red, which is neutral. Distillated water and human blood is basically, and human saliva is basically a little bit acidic. Yes. And then we got here black coffee is about 5, tomato juice about 4. That was our examples to calculate the hydrogen ion. And then we have some of other stuff here that perform a different category of pH. So let's take a look a few more, and then we coming back for the more examples. So hydrochloric acid basically very acidic and the pH about 1 here. And as opposed to sodium hydroxide on the other extreme, it's very basic. So it's a tell about the acidity and the alkalinity depends on the hydrogen ion concentration relative to the hydroxyl ion concentration. So just a comparison, it's very important to know what a buffer solution is. A buffer solution is a substance that minimizes change in pH. Its pH changes very little when a small amount of strong acid or base is added. So in the solution, if we add a little amount of even strong acid or base, they can maintain, they can buffer the pH. That's why it's called buffer solution. In aqueous solution, consisting of a mixture of weak acid and its conjugate base, or vice versa, this is a buffer solution. So buffer accept hydrogen ion when it is in excess and donate hydrogen ion when it's depleted. One good example in our system or in a body system is bicarbonate buffer. It's a primary buffer system. In our blood, we have abundance of bicarbonate ions that can maintain our blood around 7. So we're going to see more examples in the field of agriculture. Dr. Jim will speak about that in the management of this pH and so forth. So this is one important application in our system in chemistry, especially in physiology of plant, photosynthesis and respiration, how actually the production of energy during the respiration and photosynthesis really depends on hydrogen ion concentration. We're going to see here, this is a mitochondria cell membrane. The membrane is embedded of the system here, but we can going to see how ATP is synthesized by the transferring the hydrogen ion concentration from inside of the mitochondria to the lumen or to the outside of the inner membrane of mitochondria. So here's the matrix, it's abundant of hydrogen ion, and the hydrogen ion can be pumped out, make a different gradient concentration, and it creates energy, and that energy is utilized to create adenosine triphosphate. So we're going to see a video, so no worry. So, and on the other side, a photosynthetic reaction also perform almost the same thing. So when sunlight bombarded the chlorophyll, sap that electron, and then it can repel a lot of hydrogen from the water. So here, if you see, when hydrogen water is got split because of the energy of the sun, it releases a lot of hydrogen, and it makes a strong hydrogen gradient concentration, and it can pump out through this enzyme called ATPase to create ATP. So almost the same mechanics using the hydrogen ion concentration, the cell can harvest energy for work. There's one more video we're going to take a look about how this hydrogen ions gradient concentration is basically very important for life.
Concentration gradients are a key component of the biological world. The potential energy from these gradients is often used to perform biological work. Here we will focus on hydrogen ion concentration gradients. Hydrogen ions are also known as protons. A gradient exists when there is a higher concentration of a molecule in one compartment compared to a neighboring compartment. This animation will demonstrate how the potential energy that results from a hydrogen ion gradient uses ADP and inorganic phosphate, also known as PI, to synthesize ATP. This process involves an enzyme complex called ATP synthase. Gradients and the potential energy they create are key aspects of the biological world. A good example of the use of a gradient occurs in the mitochondria when ATP is synthesized. ATP is synthesized by ATP synthase, a large complex of membrane-bound protein. Here we see ATP synthase, along with other membrane-bound proteins. Notice the large difference in the number of hydrogen ions on the two sides of the membrane. This difference is a hydrogen ion, or proton, concentration gradient. The energy associated with this gradient is used to synthesize ATP from ADP and PI. This occurs at the ATP synthase complex. One hydrogen ion enters the ATP synthase complex from the intermembrane space, and a second hydrogen ion leaves it on the matrix space. The upper part of the ATP synthase complex rotates when a new hydrogen ion enters. Once 3 protons have entered the matrix space, there is enough energy in the ATP synthase complex to synthesize 1 ATP. In this way, the energy in the hydrogen ion gradient is used to make ATP. Now let's watch the process again. Notice how the proton enters the ATP synthase and exits into the matrix space. Once 3 more hydrogen ions have crossed the membrane, another molecule of ATP will be made. In this example, the hydrogen ion gradient is large enough to produce 6 ATP molecules. Please watch as the remaining ATP molecules are synthesized. The process has now completed, and the result is an equal number of protons on each side of the inner membrane. Without a gradient, there is no more energy available to make ATP. In biological systems, however, a gradient is always maintained. The mitochondrial hydrogen ion gradient is generated as electrons pass through three membrane complexes. That process can be seen in the mitochondrial electron transport chain animation.
All right, that was the video. That was very impressive, basically, because we can see the importance of the hydrogen ion to create life. One single proton actually very responsible to create life in form of energy. So let's now we move a little bit to the next part, plant nutrient availability. So the availability of some plant nutrients is greatly affected by soil pH. So now we see the connection about pH and the nutrient availability. So the ideal soil pH is close to neutral, and neutral soils are considered to fall within a range from a slightly acidic of 6.5 to slightly alkaline at the pH of 7.5 is in the range of neutral. It has been determined that most plant nutrients are optimally available to plants within the 6.5 to 7.5 pH range. Plus, this range of this pH is generally very compatible to plant root growth. Nitrogen in this regard and potassium and sulfur are major plant nutrients that appear to be less affected directly by soil pH than many others, but still are to some extent. So they affected very minutely with the pH. So on the other hand, phosphorus P, however, is directly affected. At alkaline pH value, which is greater than 7.5, for example, phosphate ions tends to react quickly with calcium and magnesium to form less soluble compounds. At acidic pH value, phosphate ion react with aluminum and iron to again form less soluble compound. So most of the other nutrients, especially micronutrients, tend to be less available when soil pH is above 7.5, and in fact are optimally available at a slightly acidic pH, for example, 6.5 to 6.8. The expectation is molybdenum, which basically they are available under acidic pH and more available at moderate alkaline pH value. So here's the diagram showing that the availability of elements as compared to as pH increase here. So here in this picture, we can see calcium and magnesium. They are, as the pH increase and the availability also increase at a point around neutral, 7.5. Since nitrogen, sulfur, and potassium almost behave in the same way with calcium and magnesium. But if we take a look here, the green stuff here, phosphorus actually become more available in the pH around 6.5, and the availability decrease when the pH actually increase. On the other hand, we see that the micronutrients is more available a little bit in the pH, acidic pH or lower pH, and it become not much available in the higher pH, except for molybdenum. So in molybdenum, as the pH increase, the availability also increased at the point until it reached 7.5. This is one way to see. And another diagram is actually the same thing. We can take a look here, that mostly the micronutrient, nitrogen, phosphorus, potassium, sulfur, calcium, they are basically available in the pH around 7 to 7, say like 6.5 to 7.5. But this iron is actually a little bit acidic. And then most of these micronutrients available in the pH around 0.5, 4.5 to 6, but they're not much available in higher pH, except for molybdenum. We have discovered that. So I think we have seen this slide from Dr. Jim. So as the pH increase, so more cation actually become available in the soil particle. So we move for the second part, which is nitrogen cycle in nature. We're going to be very brief because Dr. Jim will talk about this management part of this nitrogen quite extent. So in general, the nitrogen cycle has 5 steps. First, nitrogen fixation, which is to change nitrogen gas become ammonium by a symbiotic bacteria, for example, Rhizobium anabaena, and also non-symbiotic bacteria, nostoc. And the second process we call nitrification, where ammonium oxidized, the ammonium will be oxidized, become nitrate and nitrate by the presence of ammonium oxidizing bacteria, for example, Nitrosomonas, and nitrate oxidizing bacteria, Nitrosococcus. And the third step or part of this cycle of nitrogen cycle, we call it assimilation. It's basically incorporation of ammonium or nitrate into biological tissue, become organic nitrogen compound like amino acid and protein, nucleotide in our DNA, and so forth. And ammonification, where organic nitrogen compound become ammonia by the decomposer bacteria. So the organic nitrogen compound that we get from plant or animal is going to be decomposed by the presence of fungi and bacteria in the process what we call decomposition. And finally, we have dinitrification or denitrification, where the nitrate going back to ammonia in the presence of certain bacteria like Pseudomonas and Bacillus. So for more general or more specific, I think, we're going to see step by step of this process. Firstly, we see the biological fixation, where the nitrogen, the gas nitrogen that abundant in nature in form of gas is about 78% in nature. It can be fixed by certain bacteria. No any other organisms that actually can fix the nitrogen to become available for plants. So some nitrogen fixation is basically done by lightening strike, producing the nitrogen oxide, but most is done by diastrophic bacteria, the bacteria that utilize free gas nitrogen from the air through the presence of the enzyme that we know as nitrogenase. So nitrogen, and the enzyme is nitrogenase. So the availability depends on the microbes. There are 2 steps in basically in the nitrogen fixation. We're going to see that shortly. So nitrogen fixation and two, the nitrogen gas become ammonia or ammonium ion take place by two processes. Firstly, by symbiotic bacteria, Rhizobium and Anabaena, and many more to come. You can see in the example later. And then non-symbiotic bacteria, nostoc, and mostly cyanobacteria, they are not dependent on they are free-living bacteria that can fix nitrogen from the air. So here are some examples that we can take a look. The host-dependent and host-independent bacteria. The host-dependent bacteria, for example, Rhizobium, Leguminosarum, that means that the Rhizobium dwell in the root of the legume or the peas plant, like soybean and so forth. So this bacteria that present in the nodule actually can fix free nitrogen from the air, and then they create a molecule, what we call it ammonia. So this is when we take a closer look to the nodule. So this nodule is loaded with nitrogen fixing bacteria. So they get advantage also because they get some sugar feeds from this plant. So then after that, the nitrogenase, which is the enzyme, if you take a look here, fix the free nitrogen in form of the gas, and they convert, become become ammonium, ammonia or ammonium in the form of the ion or in the form of the gas. So that's one thing. So another thing is the host-independent nitrogen fixation, and we combine with nitrogen cycling bacteria. So the non-symbiotic nitrogen fixation include N2 fixation by free-living soil bacteria. So there are free-living soil bacteria here that can fix the nitrogen from air, and then they convert to ammonium, same process, but they are not dependent on this cell. So Dr. Jim, again, we are going to talk quite a lot about Anabaena, which actually perform this action. So another bacteria that are actually not involved in fixing the nitrogen that can convert ammonium, that is the product of this bacteria, nitrogen fixing bacteria, and convert them to nitrate and become available to plant. And some of these bacteria actually can recycle this nitrogen in a process we call denitrifying process in the presence of this bacteria, and give rise to the gas nitrogen back to the atmosphere. So this is non host independent nitrogen fixation, as along with other nitrogen processing bacteria. We're going to see a little bit extent in this diagram. So if you take a look, yeah, not really a cycle here, but it's a square, but it's in form of cycle, no worry. So atmospheric nitrogen in form of N2 can be fixed in two ways: nitrogen fixing bacteria that live in the legume roots and other nitrogen fixing soil bacteria that is host independent. So both of these bacteria can convert this free nitrogen gas become ammonium, and this can be actually further oxidized in the presence of nitrifying bacteria, and a process we call nitrification, become nitrate and nitrate. So nitrate, ammonium, basically is the most important minerals for plants. So then plants can utilize that, absorb that, and then they can create through a process called assimilation, become protein or amino acid, start from amino acid and protein to the plant. And when the plant eaten by, by animals, and they can give rise to organic material in their body, and then in their poo, it's actually that organic material can be decomposed by a decomposer fungi or bacteria and give back to ammonium. But the most important thing here that the free nitrogen can be fixed only by bacteria. That's our take-home message for today. So this, let's take a look at another video. Probably can give some impression. It's basically what I have described will be summarized in the video.
Nitrogen cycle. Nitrogen is the important essential element for all living organisms by the synthesis of amino acids, proteins, enzymes, etc. What happens if there is no nitrogen? Root systems and plant growth are stunted. All the leaves turn yellow. Decrease in crop production, soil microorganisms, life become dangerous. In atmosphere, its concentration is 78%. The circulation or cyclic movement of nitrogen from the atmosphere, physical or abiotic component, to soil, biotic component, and back into the atmosphere is known as nitrogen cycle. Nitrogen cycle involves various steps. 1. Nitrogen fixation. 2. Nitrogen assimilation. 3. Ammonification. 4. Nitrification. 5. Denitrification. Plants cannot utilize atmospheric nitrogen, so it should be converted into utilizable form such as ammonia. 1. Nitrogen fixation. This is the first step of nitrogen fixation. Conversion of atmospheric nitrogen into ammonia for living organisms is known as nitrogen fixation. This process is carried out by leguminous plants such as groundnut. Even some bacteria can fix atmospheric nitrogen. Examples of bacteria: Azotobacter, Clostridium, Rhizobium, Nostoc, and Anabina. 2. Nitrogen assimilation. The process of absorbing nitrates and ammonia into organic nitrogen is known as nitrogen assimilation. This organic nitrogen is transferred into animals' body when plants are eaten by animals. 3. Ammonification. The process of converting organic nitrogen into ammonia when animals die is known as ammonification. It is also known as mineralization. This process was carried out by some saprophytes like fungi and bacteria. Ammonia is also produced from volcanic eruptions and excretory products of animals. 4. Nitrification. The bacteria which is involved in nitrification is known as nitrifying bacteria. Ammonia is first converted to nitrites. Examples: Nitrosomonas, Nitrococcus. Nitrites are converted to nitrates. Examples: Nitrobacter. 5. Denitrification. Nitrates are converted into molecular nitrogen through nitric oxide, known as denitrification. In order to maintain the equilibrium of nitrogen in the atmosphere, bacteria which are involved in denitrification are known as denitrifying bacteria. Examples: Thiobacillus denitrificans, Pseudomonas denitrificans, and Micrococcus denitrificans. Problems with increase of nitrogen, acid rain formation, acidification of soil and lakes, increase in death of plants.
So that was the video. Now we can move a little bit about, I think from my side, it will be the last slide. Just take again a look about this nitrogen cycle in nature. Now it's really in a cycle. So we have covered 2 times from the video and from my description, basically. Again, just very briefly, I can summarize this one, that the nitrogen that can be fixed by the bacteria, they live in 2 ways, host dependent and host independent, and they become ammonia, and then convert by another bacteria, become nitrate, and this one can be absorbed by a plant to create organic nitrogen compound like amino acid, protein, and so forth. And nitrogen can be also become degraded, become a process called denitrification, and give rise to a free nitrogen in the air. So this is our nitrogen cycle for today. Dr. Jim will talk a lot about the management, about nitrogen. I think I can give the time to Dr. Jim now. Dr. Jim, you can take click to the presentation for the next topic. Do you want me to unshare the slide?
Yes, go ahead.
So I just
next slide.
Oh, you go next slide. Okay.
Okay, so we have now heard some of the basic science of acids and bases, and also you've been introduced to a macronutrient in agriculture, which is nitrogen, and the nitrogen cycle, which is a very well understood and very important body of science with respect to both plants and animals in agriculture. First, we're going to talk a little bit about managing pH on the farm. Next slide. The business of managing pH on agricultural lands is actually a process of adding a buffering agent. One part of that process is adding a buffering agent, which is called lime. Over time, agricultural lime can drastically improve soil conditions, leading to greater yields and greater profits. Next. Liming. Some people call this liming. Liming is the application to soil of calcium and magnesium, rich materials in various forms, including marl, chalk, limestone, burnt lime, or hydrated lime. In acid soils, these materials react as a base and neutralize soil acidity. So liming, all over the world, when we're dealing with acid soils, which are very common, especially in areas which get more rainfall, the liming of soils to combat acidity is a very common practice and actually quite mandatory with respect to the performance of a crop. Next. There are multiple ways in which acidity actually increases in an agricultural field. What we have here is a demonstration. The main pathways showing the involvement of nitrogen fertilizers in soil acidification. So nitrate fertilizers contribute by increasing the availability and releasing hydrogen ions into the soil solution. these hydrogen ions accumulate on the cation exchange surface, especially on the clay particulate in the soil texture. And pH will move in the direction of greater acidity with the regular and excessive use of nitrate fertilizers and ammonium fertilizers. So nitrate fertilizers might include the delivery of something like urea. Ammonium fertilizers might be potassium phosphates, ammonium phosphates is an example of an ammonium fertilizer, very commonly used. MAP, you may have heard it globally as MAP, monoammonium phosphate. So these contributions, these agricultural fertilizers used regularly over time as the nitrogen source will actually modify your pH and make the soils more acidic. Next. So this is an illustration of what it looks like when you have a system of agriculture where you're harvesting crops and raising animals. Okay, on the left side, you see this plant, which is a corn plant. It's returning to the soil, and this delivers alkalinity. It delivers the minerals in the plant back to the soil. Okay. On the right side, you see the plant is being removed. Both the corn might be removed, and in the case of producing silage on a farm, when the corn is removed, not only do you take the grain, but you take the whole plant. And in this case, this leads to acidification because the minerals in the soil that are associated with alkalinity decrease, which leads to an extra concentration of hydroxide molecules. And in this case, you will have an increase of acidification. So removing the alkaline components of the soil, the mineral components of the soil, will increase the acidity of a field. Okay, going on to the next slide. Why soils become acidic? Most soils become acidic due to leaching. Leaching is a word that's used in agronomy to describe the infiltration of mineral components in the soil. It can include nitrogen, especially nitrogen. Any water-soluble component in the soil can be leached into levels of the soil which are lower than the root zone. This process is called leaching. And excessive amounts of rain can produce this process. Excessive amounts of irrigation water can produce this same process. Sometimes there are functional reasons why we want to use leaching in agronomy, but mostly in the case of our conversation today, we'll talk about leaching as a negative in agronomy because it tends to make the soils more acidic. When soils are leached, soil cations are leached through the soil profile. Favorable soil cations are replaced by aluminum on the cation exchange site, CEC sites. When pHs are less than 4.7, aluminum is toxic to most plants. So under acidic conditions, as soils are leached and the cations are removed from the soil or they're pushed down below the root zone, the pH in the root zone can become more acidic, aluminum can accumulate on the cation exchange sites, replacing the soil cations, and aluminum is toxic to most plants. I'll tell you an interesting point here. If you do not manage your soils properly and you're growing crops which are grown under low pH conditions, and aluminum is more available in the soil solution as a result of that, those crops that you harvest will have excessive amounts of aluminum in the plant, in the grain. When you feed those plants to animals, there is a risk of aluminum toxicity to the animal. That aluminum will accumulate in the liver and it will be completely destructive to the liver. The liver tissue will break down in the presence of aluminum. I've seen this. I have owned hundreds of cows in the past. I've seen this specific problem on very specific farm sites where aluminum was very much in the soil solution. And it was also precipitating into the water table and was being pumped out through the wells. All a byproduct of mismanaging the soils on the agricultural site in the farming process. Areas receiving greater than 30 inches of rain per year are at high risk for acidity if they are not managed. Next slide.
One second, Jim. It seems like freezing. Yep.
Okay. So here is just a very simple illustration of the difference between a pH of 5.1 and a pH of 4.0 and the growth of these plants with respect to their root development. And in the case of 5.1, aluminum was at 2 milligrams per kg of in the plant. In the case of the lower pH, aluminum was at 15 milligrams per kg. You can see the difference in the plant response. Aluminum is toxic to the plant. And that's the purpose of this illustration. And it is an illustration of the points that I was making to you about the toxicity of aluminum, the danger of aluminum toxicity, the mismanagement of pH in an agricultural setting with regard to plant performance, but it's also closely related to animal performance and animal health. Next slide. Buffering pH with lime. Okay, so we have in this triangle, we have 3 levels of soil: low, moderate, high. Okay, so in the case of a sandy soil, which is at the upper part of the triangle, versus the middle part, which is a loam soil, versus the lower part, which is a clay soil with higher organic matter. On the right, you can see at the upper part of the triangle, it only takes a small amount of lime as an amendment to the soil in order to change the pH. On the other hand, in a clay soil with a high amount of organic matter, it takes a larger amount of lime as a soil amendment to change the pH. This is important to understand when you approach the process of liming your soil to manage pH. You need to understand not only the texture of your soil, but you need to understand the organic matter contents of your soil. And this is why we do soil testing. So we know how to better manage soil characteristics such as pH. Managing pH for a farmer is the application of chemistry in agriculture. Next slide. This is a schematic look at the cation exchange. So on the left, we have a high cation exchange of 25. On the right, we have a low cation exchange. And if you look at the bottom, some of the practical considerations here. Soils with a cation exchange in the range of 11 to 50, we're going to focus on one of those things. Number two, more lime is required to correct a given pH. These soils tend to have a higher clay content. On the right-hand side, when the cation exchange in the profile of your soil is 1 to 10 range, typically these soils have a higher sand content. The nitrogen and potassium leaching is more likely. Less lime is required to correct a given pH. So this gives you a measurement of soils in the form of cation exchange capacity, which is a regular feature on a soil test result. And this gives you a management guide as to how you approach the process of changing the pH as a reflection of your soil cation exchange capacity. And your soil cation exchange capacity will be different relative to the texture of your soil. Texture of your soil is the percentage of clay, silt, and sand in your soil profile. Next slide. Descriptive terms commonly associated with certain ranges in a soil pH. And I will go through them rather quickly. Extremely acidic, like in these examples we see here, and you can read the examples: lemon, vinegar, stomach acid. Very strongly acidic, moderately acidic, slightly acidic, neutral, slightly alkaline, moderately alkaline, strongly alkaline, and very alkaline. Okay. You see lime is at the end, number 12. So it is a very strong base. And as a very strong base, as Dr. Winu indicated, when you have a base and an acid interact with each other, they begin to approach the neutral pH range. So now you can see why lime is used as a soil amendment to correct soil pH when the soils are very acidic. Next slide. So let's play this slide now, which introduces the role of calcium, lime being a source of calcium when added to the soil profile. Go ahead.
One of the most important things you need to look at in your soil is calcium. What is your calcium level in terms of both parts per million and percent base saturation? We're going to talk about the importance of calcium today.
Calcium is really important for the structure of your soil. You need enough calcium out there to have good pore space, to have good microbial life, good air movement, and also good water infiltration through your soil. Calcium is really important for nutrient uptake as well. In fact, it's been said that most nutrients come in on the back of calcium into your plants. You need calcium to be able to open that plant up and allow those nutrients to get in. So it's one of those really critical nutrients. However, for most farmers, they're looking at N, P, and K. How much nitrogen do I need? How much phosphorus do I have to apply? How much potassium do I need out in my soil? And they're kind of forgetting that the first thing they should be looking at is how much calcium I've got.
One of the things Darren didn't mention with calcium is soil pH. If you have a low soil pH, the fix to that is quite simple. It's lime. It's calcium carbonate. Now, you want to be careful about this because if you put on too much calcium carbonate, you can actually raise the pH beyond where you want it to get to. And also, you've got to look at what crops are you raising. So, for example, if you're raising blueberries, well, blueberries like a 5.5 pH. Corn likes about a 6.2, 6.3, 6.4. Okay, if you're raising alfalfa, that prefers somewhere around a 7 pH. So you might want to vary this depending on your crop and your cropping rotation. But again, calcium is tremendously important when it comes to pH.
One of the things that you want to look for on your soil test is your base saturation test. And if you don't have a base saturation on the soil test that you've been sending in, we'd strongly encourage you get a complete analysis done where you have micronutrients and base saturation and more to look at. With base saturation, there are 5 nutrients that we're looking at: calcium, magnesium, potassium, sodium, and hydrogen. And when we look at calcium, this one should occupy 65 to 75% of that base saturation test. If you fall somewhere in that range, your pH is generally going to fall in line, or at least relatively close, and you're going to have enough soil porosity to be successful with what you're doing with crop production. If you're below 60% calcium, in many cases, what we would ask is that you put some lime out there to try to raise that calcium up. Those 5 nutrients in base saturation add up to 100%. And if we're low in the calcium, we must be high in something else.
Now, when Darren talks about 65 to 75%, let's say you have 90 or 95% calcium. Look, we're not telling you that in one shot, in one application of a bunch of other stuff, you can drop that calcium down to 75% and make it pay. Now, I'm not saying that wouldn't necessarily be a good thing for you to get the calcium down a little bit, but it's going to cost some money. You'd have to add some other products to it, and it really could take some time. So the big thing to focus on here is let's take a look at soil pH, let's take a look at parts per million of calcium, and ideally, yes, if we're in that 65 to 75% range of calcium, that's great. If not, we should probably strive toward that range as a long-term goal on our farm, and usually we're going to have healthier soil.
So to sum things up with calcium, it's important to look at the parts per million. It's also important to look at the base saturation percentage so you can start to determine what's going on, what am I high in, what am I low in, and do I need to add more calcium to help my crop. One other thing that will help your crop is controlling our weed of the week. Can you identify this week's weed?
Okay. So this next slide is a presentation of soil pH ranges. And Dr. Winu touched on this in presenting the mathematics associated with pH. And just note that starting at neutral, you'll see that going from a pH of 7 to a pH of 8, the soil becomes 10 times more alkaline. Going from 8 to 10 is another multiplication of 10, and it just keeps multiplying by 10. Acidic in the opposite direction is the same simple multiplication, each time multiplying by 10. So even though the range is 0 to 14 on a pH scale, it's important to be sensitive to the fact that when you move on the pH scale 1 point, let's say from 7 to 6, or you move from 6 to 5, the degree to which the soil is becoming more acidic, from a mathematical point of view, it's quite dramatic. It's quite dramatic. So the plant's response to pH in a soil is quite sensitive. And if you get off the scale very much, even though it may not seem like a big move from 7 down to 5, the degree of acidity is 100 times greater at 5 than it was at 7. And this is related to the hydrogen ion concentration, which is the chemistry driving a pH measurement. Okay, moving on to the next slide. Now we're going to jump in the opposite direction. We've been talking mostly about acidic soils. Now let's talk about an alkaline soil. Now, alkali or sodic soils is defined as a soil having a conductivity of the saturation extract less than 4 ds over m. The formula is not too important right here. And an exchangeable sodium percentage, ESP, greater than 15. That is a very important number and is the number that you're most likely to see on a soil test, is the percent of exchangeable sodium. 15 would be considered a high number. The pH is usually between 8.5 and 10. That's the pH measurement. Formerly, these soils were called black alkali soils, and the soil is called solid, or degraded alkali or sodic soils. The term that I'm most familiar with is sodic soils. Alkaline or saline soils. Next slide. In the United States, we have a fairly simple representation here of what the United States looked like with regard to acidity versus alkalinity in soil characteristics. You can see in the western part of the United States, approximately half of the country, we have more alkaline soils. We have drier climates. We have more porous soils. We have higher sand contents in those soils. They tend to be alkaline by nature. In the western part of the United States, there's very little liming of soils. On the opposite side, the eastern half of the United States, more higher quantities of rainfall normally, and you have more acidic soils normally. And in terms of agricultural industrial practices, this is the part of the United States where you'll see the use of lime to control acidity. Next slide. Okay, so we have presented this information a few times now about the availability of nutrients with regard to acidic versus alkaline conditions. You'll see that that range of 5.5 up to 8 will accommodate most plants. Now, there are plants that are more tolerant of acid conditions, and you might even want to create a pH of 4 in your field soil profile in order to accommodate a plant that is highly tolerant of acid conditions. We might say it likes acid conditions. We might say that it grows best under acidic soil conditions. A blueberry plant is an example of that. On the other hand, there are plants that are tolerant of alkaline conditions. And most of those plants will be in the 8 to 9 range. Once you get over 9 or 10, then it's hard to find plants that are very tolerant. But under more alkaline soil conditions, you will have certain types of plants that are better adapted. Cotton, for example, will tolerate alkaline conditions. Cotton is commonly grown in the western part of the United States, where soils are naturally more alkaline. Next slide. Alkaline soils are neutralized with the addition of elemental sulfur, iron sulfate, or aluminum sulfate. And there are other acids that are often used to neutralize alkaline soils that's not mentioned here. One of them is phosphoric acid. Phosphoric acid is a source of the macro element phosphorus. And phosphoric acid is used in states like California in the western part of the United States in order to combat alkalinity in the soil profile, as well at the same time to deliver phosphorus, a macronutrient for crop production. But sulfur-related chemistries are used to combat alkaline soil conditions. Next slide. Farmers manage pH. They manage acid soils. They manage neutral soils. And that may seem odd to you, But sometimes it's important to make the soils more alkaline. Sometimes it's important to make the soils more acidic in order to accommodate the crops that you're growing. On the other hand, I would suggest to you that it might be smarter and more intelligent to select crops that are best adapted to the natural conditions of your soil in the ecosystem that you find on your farm and the climate that you live in. Also, farmers have to manage alkaline soil conditions as well as saline soil conditions. Saline soil conditions are those soils that are high in sodium. Gypsum is used to reduce the amount of sodium that is exchangeable in the soil solution. And gypsum is used with large amounts of irrigation water in the western part of the United States to reduce sodium levels in soils. And the irrigation water in large amounts is used to leach the exchangeable sodium. The gypsum supplies a source of calcium, which replaces the sodium on the exchange site. So the soil moves from being more saline to being more alkaline. Okay, moving on to the next part. We're going to make a few brief comments here about the nitrogen cycle. You've already been presented with much of this information. I'm not going to repeat in detail what this is, but we're going to move a little bit more into managing nitrogen on farms. Next slide. So we're going to focus largely on two elements in nitrogen management on farms. And the motivating reason for doing this is we're going to look at the biology of nitrogen fixation. And what motivates us about this is the fact that the most common source of nitrogen on the planet is in the atmosphere. As Dr. Winu presented, nitrogen in the form of a gas, N2, makes up 78% of the atmosphere. And notice that this atmosphere is available to everyone at every location across the entire planet. Accessing nitrogen for agricultural purposes from the atmosphere is a relatively free process. In contrast to using hydrocarbons or petroleum and natural gases to produce chemical sources of nitrogen, which are, you might think of as synthetic sources of nitrogen, which have to be transported around the globe in very large quantities, in bulk, which consumes a lot of fuel. They are not uniformly available across the planet, whereas the atmosphere is available. So in agricultural management, in crop management, it's important for the operator to understand that freely available to them is atmospheric nitrogen. The reduction of atmospheric nitrogen to a plant available form of nitrogen is the nitrogen fixation process performed by bacteria. So symbiotic roots, symbiotic relationships between a microorganism and a plant takes place in legumes, for example. Legumes like peas, peanuts, soybeans, all kinds of beans. These are all examples of legumes. Alfalfa is a legume. These microorganisms form nodules on the roots of those plants, and that's what you see photographically presented here. This is sometimes called mutualism. A mutual relationship will exist. They have an interplant biological relationship, Mutualism between a plant and bacterium, which allows for fixation of atmospheric nitrogen to form that a plant can utilize. The bacterium is rewarded with food and a place to live. And on the right-hand side, you see a cross-section of a nodule. The bacteria live inside the nodule. They receive sugars or carbohydrates from the plant, which feed their metabolic system. And as a result, they actually collect nitrogen from the atmosphere. This is taken from the soil atmosphere, the soil air. Ideally, a soil will be 25% air. And it is inside the air that's in the soil that you find N2. The bacteria interact with that air. That air moves across the membrane of the nodule. The bacteria then access the nitrogen that's in the soil air. Moving on to the next slide. This is a micrograph, very close up, of a rhizobia bacteria. Okay, this is an example of a bacteria that fixes nitrogen. Next slide. Here you see the bacteria beginning to colonize, these rhizobia bacteria beginning to colonize the growing point of a root hair. This is also a micrograph of that process. Next slide. This is looking at the same thing, but looking at a magnification of a root hair and the nodules that are beginning to form on that root hair. As the root hair expands, the rhizobia, it follows the root and will colonize the surface of that root hair, and it will tap into the sap or the solution that's inside of the root itself, and it will harvest a certain amount of carbohydrate that it might need to support its metabolic system. The nodule is the housing for the rhizobia bacteria. The soybean plant is the host. Okay, moving on. Root nodules on a soybean plant. This is what they look like. This is a naked eye view on the left. And then obviously on the right, we're looking at various 2 levels of magnification. Next slide. So there is a business in agriculture of inoculating the seeds of a legume with rhizobia bacteria. We're looking at soybean plants here. When you apply the rhizobia microbiology to the surface of the seed, it's normally applied it can be applied as a liquid or a dry material. You do it directly in the planter box when you're sowing your seed, when you're planting the seed, or it can be done at the factory where the seed is processed and put in the bags for distribution to the farmers. So getting rhizobia to the farm, put it on the seed, plant's performance will respond. This is a pot study that attempts to illustrate the plant response to seeds that have been inoculated versus seeds that were not inoculated. And you can see in this case, the plant response is quite profound. So there is agronomic value. There is economic value in farm management in utilizing the application of microbiology, in this case, rhizobia, in order to capture nitrogen from the atmosphere, deliver it to the plant itself. Rhizobia captures the atmospheric nitrogen, delivers it to the plant. plants utilize the nitrogen to form all kinds of molecules, for example, proteins. It fuels the metabolic system of the plant, and at the surface level, on the level of observation, you can see a plant's response. That's what you're looking at here. Next slide. So this is a field observation. At the top, you have soybean plants. On the right, they have been inoculated. On the left, no inoculation. At the bottom, you can see the level of nodulization that has occurred. On the right-hand side, these soybean roots are well nodulated, very large nodules, a very large population of rhizobia bacteria. On the left, virtually no nodulization. These fields need to be inoculated with rhizobia bacteria. You can see the plant response at the top. On the right, the plants have a better color. There seems to be a greater biomass density in the vegetative growth of these soybeans. The plant response will produce more photosynthate. More energy will be produced in that plant as a result of its response. It's really a response to nitrogen in the plant. And at the end result, the yield on the field where the rhizobia population is quite high, the yield will typically be greater when compared to a field without a rhizobia population on the root zone. Okay, next slide. Now we're going to shift to a host-independent organism. These are blue-green algae or cyanobacteria. Next slide. Cyanobacteria are oxygenic photosynthetic bacteria that are widespread in marine, freshwater, and terrestrial environments. And many of them are capable of fixing atmospheric nitrogen. They commonly occur in soils. That's what terrestrial environments mean. They occur on land. They occur in the soil. All that are heterocyst forming are capable of aerobic nitrogen fixation. And in the absence of combined nitrogen, 5 to 10% of the vegetative cells will differentiate into specialized cells called heterocysts that provide an environment suitable for the functioning of nitrogenase. This is the enzyme that catalyzes the process of nitrogen fixation. An example, nostoc. Nostoc is a genus of filamentous nitrogen-fixing cyanobacteria that form macroscopic or microscopic colonies and is common in both terrestrial and aquatic environments. A heterocyst is very similar to the nodule for a rhizobia in that both a nodule, the membrane of a nodule, and the membrane of a heterocyst is such that it minimizes, the membrane is structured such that it minimizes the intake of oxygen. Mm-hmm. As gases move across the membrane of a heterocyst or a nodule, they restrict the intake of oxygen. And there's a reason why this is biologically and chemically important. Oxygen destroys nitrogenase. So a heterocyst, morphologically speaking, is designed to protect nitrogenase, and it does so at the level of its membrane. A nodule for rhizobia on a host like a legume plant, that nodule has a similar function. It keeps oxygen outside. The membrane of the nodule keeps oxygen at a minimum inside the nodule. so that the nitrogenase enzyme is not destroyed. Go on to the next slide. This is a microscopic image of a cyanobacteria. And what you see there is the specialized cell that is a heterocyst. And you can notice that the cell wall of the heterocyst is quite thick. Okay, so this is just a visual observation of the thickness of the wall of the heterocyst cell, and the morphology of that cell is an important part of the chemical functionality that's taking place within the heterocyst. Nitrogenase exists within the heterocyst, within that specialized cell. And as atmospheric gases that contain nitrogen move across that membrane, those nitrogen molecules are reduced to ammonia forms. And that process is catalyzed by the enzyme nitrogenase. Next slide. So this is a graphic presentation of a cyanobacteria nitrogenase or heterocyst cell. And it's in this case, we're looking especially at the nitrogen element, which is produced especially by the heterocyst cell in a cyanobacteria organism. A very unique thing about cyanobacteria, they are photosynthetic, they're host independent, and they're generally very independent organisms. They're basically microscopic plants. They need CO2 from the atmosphere. They need H2O, which comes from the soil solution. They need light that comes from the sun. They need a small amount of minerals or nutrients, which come from the soil mineralogy. These organisms contribute greatly to the ecosystem of a soil. They are important in bioremediation, in production of contributing to bioenergy systems. They produce carbohydrates. They produce biofertilizers. Nitrogen is the one that we're focused on today, which could be called a biofertilizer in this case. Nutraceuticals, and they contribute to soil fertility. These organisms will reduce phosphate rock. They will reduce non-available forms of phosphate or phosphorus in a soil solution. They will reduce it to P2O5, which is the plant available form of phosphates in the soil. Okay, so they play many functions. The primary function that we're talking about today in talking about the nitrogen cycle and managing nitrogen on a farm is the role of cyanobacteria to fix nitrogen and to contribute that to the soil fertility. Next slide. So farmers manage the nitrogen cycle. They use biological methods, which, for example, can include the delivery of compost to a field, which is rich in all forms of biology. It may include increasing the presence of worms in the soil profile. The presence of worms in a soil profile is a very good thing with respect to soil structure, in that it aerates the soil really well. Worms are very good at that. Microbiological methods are utilized, like rhizobia and cyanobacteria. Chemical methods are utilized, like the delivery of Chilean nitrates. Chilean nitrate salts are mined from the earth in the country of Chile. They have huge deposits of this material, and it's used in organic agriculture as a source of nitrogen. Physical methods are used to manage the nitrogen on a farm. It's important to minimize the loss of nitrogen from your field by using methods under soil conservation to stop erosion. When water moves soil from your field, it moves nitrogen from your field as well. Nitrogen, many forms of nitrogen are very water soluble. The water soluble forms of nitrogen can be lost through water soil erosion. So you have to control that. And this is a physical method for managing nitrogen on your farm. Next slide. So thank you for this today. We will be back in a week to present the final session on chemistry and agriculture. So for today, we'll say goodbye and we'll see you all again in 1 week. Thank you.
Real quick, Jim, we do have a couple questions from
Okay.
some people. So let me just pull those up really quick.
I've got them here.
Okay, great.
All right. So first question, what is the primary component affecting potassium availability? So first of all, potassium needs to be in a form that is available. Potassium may be present in soils in many different forms. But potassium is only available to a plant in one form, K2O5. And this molecule can be, this form of potassium is water soluble. It can go into the soil solution. It can move osmotically across the plant root membrane, or energy can be utilized by the plant in the form of ATPs, creating a metabolic pump to force these minerals across the membrane of the plant. These are the two methods in which plants will take up a mineral from the soil solution. So first of all, in order to make sure that potassium is available to the plant, it has to be specifically in one chemical molecular form. Okay? Next question, is application of inorganic fertilizer a best way to solve the problem of alkaline soils? An inorganic fertilizer, an example of that is monoammonium phosphate, MAP. That would be considered an inorganic fertilizer. And the question is that the best way to solve a problem of alkaline soils? Okay, as you saw in my presentation, the more common ways of managing alkaline soils are the use of acids that are put into a soil profile. Now, another very interesting way of managing soils that are alkaline by nature and tend to have a high pH, whether it's driven by a concentration of sodium ions or calcium ions, both saline and alkaline conditions in a soil profile, is to use microbiology. Okay, cyanobacteria are known to produce what's called alginic acids. As these acids are released into the soil profile, they will break the bond that connects the calcium or the sodium to the cation exchange site, allowing both sodium or calcium to be moved by leaching through the soil profile. And pH will go down as a result. So using microbiology is a method that I particularly like a lot, applied microbiology to manage soil alkalinity. It's cheaper. The dosage rate is really small compared to using something like gypsum or large volumes of liquid acids like phosphoric acid. The inoculant rates for cyanobacteria in a clay carrier can be as low as 100 grams per hectare. The microorganisms grow in the soil. As the population increases, they excrete acids that combat alkalinity in the soil. So we're learning a lot in agriculture today about ways to apply biology and apply microbiology to solve agronomic problems. As we do that, we're learning to become less dependent on chemical methods to solve agronomic problems on farms. Okay? That's the answer. Okay, Kelly, so we'll finish now. Thank you
Alrighty,
all.
well, thank you so much, everybody, and we'll see you next Friday.
Thank you.
Thank you.
Bye-bye.